Now coming to the second topic of today's class, that is immunology in obstetrics. Immunology as such is a very complex subject, but we'll try to make it a little simpler. So at the end of this class, we should be able to describe basic terms related to immunology, basics of reproductive immunology, immune tolerance of pregnancy, mechanism of infection of fetus, fetal and newborn immunology, and immunological control of fertility. So let us go to the preclinical classes where we had learnt about immunity. So immunity and immune response. Immunity is the ability of an organism to resist a particular infection or toxin by the action of specific antibodies or sensitized white blood cell. So process of eliminating foreign substances by the body is called immune response, and the substance responsible is called as antigen. So antigen can be either a microorganism or some molecule in case of toxins. So the initial response to an antigen is called as primary response, which occurs in 1 to 2 weeks of an assault. So subsequent exposure to the same antigen leads to what is called as secondary immune response. So there can be antigen specific and antigen non-specific immune reactions. So the immune system is capable of differentiating between self and non-self, and the failure of this leads to autoimmunity. So different antibodies develop in response to various antigens. Generally, immune system is divided into two arms, that is innate and an adaptive immune system. So what is innate immune system? It is the first line of host defense against infection. It works at fast pace. It recognizes the pathogen. It cannot identify the self versus non-self, that is non-specific, and involves a complement system. The complement system is a part of the immune system that enhances complements, which are a type of plasma proteins. So the complements the ability of antibodies and phagocytic cells to clear microbes and damaged cells from an organism. This complement system promotes inflammation and attacks the pathogen's plasma membrane. The second arm of the immune system, that is the adaptive immune system, works as a second line defense against infection. This has a delayed response, contrary to the innate immune system. This can discriminate between self and non-self, and it prevents a reinfection. So this system has got mainly two types of cells, that is lymphocytes, which is B and T lymphocytes, and antigen presenting cells are called as APC. So there are three components of an antigen specific immunity or adaptive immunity, which include memory, specificity, and recognition. Specificity is the immunity response aided by the memory. So on electrophoresis, 5 major types of immunoglobulins are found, which are IgG, IgM, IgA, IgD, and IgE antibodies, which are involved in primary immune response. They act as cytolytic agents by activating the complement system. So immune cells involved are phagocytic cells, that is neutrophils, monocytes, and macrophages, natural killer cells or NK cells. So and eosinophils and basophils. So what is left of the white blood cells over here is lymphocytes, which are involved in the adaptive immune system. Immune responses are by the release of cytokines. Cytokines is a general term which is used for the signaling molecules. Cytokines like TNF alpha or tumor necrosing factor alpha, interleukin 1 and interleukin 6, and chemokines. Chemokines are a specific cytokines that functions by attracting cells to sites of infection or inflammation. So they are like chemotactic involvement is there. There's a chemotaxis in nearby responsive cells, and that is the chemokines are also called as chemotactic cytokines. So chemokines are interleukin 6, MIP1 alpha, and MCP1. So phagocytic cells, what do? They ingest and digest microorganisms with lysosomal enzymes, which are there already present in the cells. And natural killer cells can't recognize and distinguish between normal cells and cells infected with a virus or tumor, that is self or non-self. So there is modification of immune response system in pregnancy. So as such, pregnancy has been regarded as a immunosuppressed state with progesterone acting as an immunosuppressant. However, there is no generalized immunosuppression during pregnancy. In fact, there is a modification in immune response in pregnancy to adapt to the foreign fetal allograft. So many of the maternal immunological adaptations to pregnancy are not well elucidated. So modification can occur at various levels, that is at the level of T lymphocytes, peripheral natural killer cells, monocytes, and granulocytes. So T lymphocytes, there are two types of T lymphocytes. One is the helper T lymphocytes, it's also called as TH cells, and cytotoxic T lymphocytes are called as TC cells. So helper T lymphocytes, they produce cytokines and are further divided into type 1 cells and type 2 cells. So type 1 cells, they produce interferon gamma, interleukin 2, and tumor necrosis factor alpha or TNF alpha. These activate macrophages and are responsible for cell-mediated immunity. Type 2 cells, on the other hand, they stimulate B lymphocytes and produce interleukin 4, 5, 9, 10, and interleukin 13, and are responsible for promoting systemic humoral immunity and suppressing local cell-mediated immunity. And in pregnancy, associated with an increase in the CD4 positive T cells secreting Th2 type cytokines, and there is a shift from type 1 to type 2 cells, that is, there is a shift from cell-mediated immunity to humoral immunity, which is essential for sustaining pregnancy. So if you take this into consideration, of the various autoimmune diseases, in cases of rheumatoid arthritis, which is a cell-mediated autoimmune disorder, this undergoes remission during pregnancy, while systemic lupus erythematosus, which is an antibody-mediated immune response, this disease worsens during pregnancy. So Th2 shift is influenced by hormonal factors, that is progesterone, and release Th2 stimulating cytokines by macrophages. So this bias towards Th2 affects the ability to rapidly eliminate certain intracellular pathogens during pregnancy, although the clinical implications of this suppression are unknown. The other cells are cytotoxic T lymphocytes or TC cells that directly attack the foreign bodies or infected cells and destroy them. So a few words about the dendritic cells, which are also to be taken into consideration when we are considering immunology of pregnancy. They are antigen presenting cells, also known as accessory cells of immune system. So this we had dealt with when we were dealing with the innate and adaptive immune system. They act as messengers between the innate and the adaptive immune systems. So sorry. So the dendritic cells, their main function is to process the antigen material and to present it on the cell surface to the T cells of the immune system. So in pregnancy, the dendritic cells are thought to play a crucial role in mediating the balance between the immunity and tolerance and determining the type of T helper cell response. So this is one of the representation of T cells during pregnancy. It is so this is a 10 to 20% of the leukocytes in the first trimester are said to be CD3 TCR. That is a cluster of differentiation 3 and T cells co-receptor that is involved to generate an activation signal in T lymphocytes. So it's a complex of CD3 and TCR or the T cell receptor. So this is again divided into, of this there are 30 to 45% are CD4 that helper cells and and a small number are mucosal T cells expressing T cell receptor and the natural killer cells. Then there are T regulatory cells also in this of the CD3 TSR complex. So CD4, which constitutes the 30 to 45% of this CD3 TCR complex, in that one 50% are due to memory and 5% are regulatory, and in CD8 complex, which constitutes 45 to 75%, it is due to memory. So CD8 are cytotoxic T lymphocytes and CD4 are the helper T cells. So the Th2 cells, so in cases of spontaneous abortion and preeclampsia, there's a decrease in the TREX or the T regulatory cells and increase in the Th17 cells. And Th1 cells, which we had dealt earlier, that is which causes cell-mediated immunity, they called recurrent spontaneous abortions. So other modifications like peripheral natural killer cells, they have a deleterious effect on the pregnancy and their number and function usually suppressed in pregnancy. So they are capable of killing target cells and that are coated with antibodies. Excessive presence of peripheral natural killer cells is associated with miscarriage. So in pregnancy there is an unusual kind of decidual natural killer cells which perform a developmental role in humans. In preeclampsia, there is impaired activation of natural killer cells, and that's why there is a uterine vascular remodeling during pregnancy, which will be shown in the next slide. So then there are monocytes and granulocytes. During pregnancy, they phagocytose fetal cells in maternal circulation. So here innate immunity gets activated by hormones and the placenta. So this diagram you must have seen when we are dealing with preeclampsia, what happens during preeclampsia and why there is a decreased uteroplacental transfusion. So in this one there is normally what happens is there is a uterine vascular remodeling during pregnancy. So this trophoblasts, they will attack the spiral arteries, so the blood supply increases as the pregnancy advances, whereas in the preeclampsia this activation is suppressed, so the spiral arteries remain narrow, and that's why the blood supply to the at the maternal-fetal interface is hampered, and that's why the various adverse this thing changes occurs in fetus. So natural killer cells or the decidual natural killer cells functions are it transforms the spiral arteriole in decidual sorry replacing endothelium by the trophoblast. There is an incomplete spiral transformation and failure of the trophoblast in preeclampsia and intrauterine growth restriction. There is a role of interferon gamma in remodeling of spiral arteriole. So if there is an inhibitory signal to the decidual natural killer cells, this results in recurrent spontaneous abortion and IUGR. If there is a trophoblastic endovascular migration, then this affects the decidual natural killer cells and causes hypoxia. The role in the intrauterine, there is a role also in intrauterine inflammation and abortion, so which causes release of cytotoxic granules, which are perforin and granzyme. So natural killer cells, the balance between the is very important in sustaining the pregnancy. So role of lymphocytes and monocytes. Immune system as such develops early in the fetus. B lymphocytes appear in fetal liver by 9 weeks and in blood and spleen by 12 weeks. T lymphocytes begin to leave thymus at approximately 14 weeks of gestation. And in the newborn, monocytes are able to process and present antigen when tested with maternal antigenic specific T cells. Despite this, the newborn usually responds poorly to immunization. So immunology in pregnancy, and that is how the immune tolerance occurs, why the fetal allograft is not usually rejected. So immune tolerance of normal pregnancy at the maternal fetal interface is maintained by the interaction of human leukocyte antigen G with the uterine natural killer cells. This effect predominates in a normal pregnancy. So, and levels of complements and cytokines are often raised during pregnancy. However, there is the inhibition of such complements and cytokines by the placenta. This in turn reduces the immune-mediated pregnancy complications. In the absence of the direct antigenic stimulus, such as infection, fetal plasma immunoglobulins consist almost totally of transferred maternal immunoglobulin, that is IgG. The antibodies in the newborn are most of the reflective of the maternal immunologic response. So there is lack of uterine transplantation immunity during pregnancy is unique. So some note the immunological peculiarity of the cells involved in implantation and fetal placental development. These include decidual natural killer cells. So during pregnancy, trophoblasts are the only fetal derived cells in direct contact with the maternal tissue. So that is the very important barrier between the mother and the fetus, whether it can remain as an ongoing normal pregnancy or it can have an adverse effect on the fetus. So decidual natural killer cells in pregnancy, they usually mediate angiogenesis. So residual natural killer cells work in concert with the stromal cells, and they produce pro-angiogenic factors during pregnancy which help in sustaining the normal pregnancy. So then there is supposed to be a role of blocking antibodies in sustaining the pregnancy. In 1960, it was suggested that the fetuses that shared different paternal and maternal antigens has selective survival advantage over those sharing similar antigen. So it was assumed that the mother produced a blocking antibodies to fetal foreign bodies antigens in pregnancy. The failure to develop blocking antibodies in cases of similar maternal and paternal antigens would result in a reproductive failure. So that was the concept earlier thought of, but this concept has however been challenged and doesn't hold true anymore. Until date, there has been no standardized assay for determining the presence of these blocking antibodies. So protection of the concept is, we already dealt with a few concepts, and there is, besides that, there are several local and systemic modifications are now suggested to be involved in protection of the developing fetus from attack by the maternal immune system. So the protection are by the three main mechanisms which are there besides the others are cytokine shift, influence of the female sex hormones, and a unique human leukocyte antigen expression by the trophoblasts. So in successful pregnancy is associated with the dominance of Th2 type immunity, which we have dealt earlier. That is a hormonal mediated immunity, and induction of Th1 type responses is considered potentially dangerous to the continuation of the pregnancy. So this paradigm of cytokine shift has been expanded to consider T regulatory cells and the Th17 cells, with T regulatory cells playing a pregnancy protective roles and then Th17 responses being detrimental to pregnancy. This we had seen when we were dealing with the T cells. An influence of the female sex hormones, that is the estrogen and progesterone levels, are massively upregulated during pregnancy, and both hormones have been shown to have immunomodulatory function, impacting immune cell recruitment, expansion and function. So they are for the normal continuation of the pregnancy. And unique human leukocyte antigen, the trophoblasts are the main cell type of the placenta, and they exhibit unique major histocompatibility complex expression compared to the mother and baby. So the major histocompatibility complex 2 molecules are not expressed by trophoblast, and HLA of the major histocompatibility complex type 1 are expressed by trophoblast and have functions including control of depth of trophoblast invasion and binding of natural killer cell receptors. So these are the various factors which are responsible for immune tolerance. Let us summarize some of them. The pregnant uterus is considered to be an immune privileged site. So what are the various protective mechanisms? There is a lack of major histocompatibility complex class 1 antigen expression by autotrophoblastic cells. There is a modification of immune system at peripheral level, that is shift to humoral immunity, that is the to Th2 cells, and cytokine shift from the pro-inflammatory to anti-inflammatory type. Then there are HLA blocking bodies, so that has been not proved as such by many. And then there is a favorable decidual changes. Then fast-fast ligand system is one of the other factor. There's a genome alteration. Then the favorable endometrial changes during implantation. So these are the various factors which are responsible for the immune tolerance during pregnancy. So in the implantation, which is the event to be considered when the rejection can occur, the salient points of this is this implantation is the result of a two-way dialogue between the embryo and the mother, and this is modified by various chemicals, cytokines, enzyme, and these are in turn under the control of hormones and the genetic factors. So it is imperative that the right amount of endometrial digestion is accompanied by the right amount of cytotrophoblastic invasion or burrowing and vascularization. Too less and too much, both are harmful for continuation of the pregnancy. So all this requires a host of physiological changes in the mother which need to be fine-tuned. So these changes, if not cleverly regulated, could be a source of potential harm to either mother or to the fetus. And in the majority of cases, fortunately, the changes proceed under the perfect balance without even being noticed clinically. So the right balance of the hormonal milieu as well as the right balance of the cytokines, natural killer cells, sulfhydryl groups and the matrix metalloproteases in the endometrium is required for successful implantation to occur. Fetal antigens, trophoblast and maternal immune system, they all work in harmony with each other for the successful outcome of pregnancy. So this is somewhat in summary of the immune tolerance. So the trophoblast, the decidual nuclear natural killer cells interaction, the interaction between the both is essential for the success of pregnancy. Threat in reproductive success is due to pro-inflammatory molecules of macrophages, which is interleukin 6 and TNF alpha, and decidual macrophage exposure to colony stimulating factor 1 is a major mechanism for local monocyte recruitment. Paucity of dendritic cells limit adaptive T cell responses and minimizing immunogenic responses to fetal placental antigens. Impaired activation of immune cells lead to pregnancy complications like preeclampsia, IUGR and abortion. And decidua should be considered as ground zero for avoiding immunological assault and during infection. So this we can skip. Then the infectious disease control. The organisms infect placenta and amniotic membrane. Like in the case of P. falciparum infection, it is the maternal blood which is affected, and in turn the villus tree is affected. And in other organisms, the decidua, which is the ground zero for avoiding the immunological assault. Then it affects the placenta and results in these various factors, that is the preterm birth, IUGR, stillbirth and congenital anomalies. So in case of this is the description of acute chorioamnionitis, where there is a interleukin 17 and 22 are involved, and these stromal cells are involved, and then this is toll-like receptors. There is a class of proteins that play a key role in the innate immune mechanisms, and this in turn affects the neutrophils. So there is an ascending bacterial infection from the cervix, and this usually causes the preterm birth, and neutrophils are recruited to fight this infection. So a word about inflammatory markers, since we are considering infection. So many tests are performed to diagnose inflammation during non-pregnant state, cannot be used reliably during pregnancy. Or the median values of C-reactive proteins are higher. ESR is also increased in normal pregnancy. Complement factors C3 and C4 are significantly elevated, and levels of procalcitonin, which is a precursor of calcitonin, increases the end of the third trimester of pregnancy. The procalcitonin levels are elevated in severe bacterial infection but remain low in viral infections and non-specific inflammatory response. And the fetal and newborn immunology. Active immunological capacity of the fetus and newborn is compromised as compared to the children and adults, and fetal cell-mediated antihumoral immunity begins to develop by 9 to 15 weeks of gestation, and the primary fetal response to infection is immunoglobulin. Passive immunity is provided by IgG transferred across the placenta from the mother, whereas the IgA cannot be transferred from the mother to the fetus. By 16 weeks, transfer of IgG across the placenta begins to are equivalent to those of the mother, and bulk of the IgG increase is during the last 4 weeks of the pregnancy. After the birth, so that's why since this is the factor which occurs the last 4 weeks of the pregnancy, preterm neonates are endowed relatively poorly with protective maternal antibodies, and that's why they are more liable for infection. And newborn begins to slowly produce IgG, and adult values are not attained until the 3 years of age. And after the birth, breastfeeding is protective against some infections, and this protection begins to decline at 2 months of age of the neonate. The baby. Then a few words about the immunoglobulin M and A. In the adult, production of immunoglobulin M is in response to an antigenic stimulus is superseded in a week or so predominantly by IgG production. Very little IgM is produced by the normal fetuses. So with infection, IgM response is predominant in the fetus and remains so for weeks to months in the newborn. And any IgM found in the fetus is that which is produced by it, not transferred from the mother. It is the IgG which is transferred from the mother. So immunoglobulin A ingested in the colostrum provides the mucosal protection against enteric infections. So now immunological control of fertility, it involves placental hormones, human sperm antigen, zona pellucida, and anti-sperm antibodies. So in immunologic placental hormones, it's the human chorionic gonadotrophin which cross-reacts with the luteinizing hormone TSH and FSH and has a beta subunit that is non-immunogenic. And there are many anti-HCG vaccines are available, but most effective anti-HCG vaccine is the one that is directed against the C-terminal peptide on the beta subunit of HCG. This antibody does not cross-react with the LH and thought to be effective up to 12 months. In the human sperm antigen, there are numerous sperm antigens present, and antibodies against the lactate dehydrogenase isoenzyme, that is X, is being currently studied. There is a significant reduction of viable sperm and improper implantation of the ovum in experimental animals. The anti-LDHX antibody is injected into them. Other factors related to fertility is zona pellucida, which is the one which is most extensively studied potentially oocyte target antigen. Anti-sera to it blocks sperm penetration as it coats the zona surface. So sera from the infertile women with anti-zona pellucida antibodies can block in vitro fertilization and accounts for failure of this process. So anti-sperm antibodies like IgG, IgM or IgA present either in the serum or in the reproductive tract. Antibodies present in the female reproductive tract that binds the sperm surface antigens affect the motility of sperm and may cause infertility. So these are the various complex mechanisms of immunology of fertility, immunology which happens during pregnancy and which sustains the pregnancy for 9 months till delivery. Thank you.